Good morning, everyone. Respected Chief Guest, Principal, Vice Principal, other dignitaries, dear faculty and students. On behalf of Department of Pharmaceutical Chemistry MCOPS, I welcome you all to this first lecture held in commemoration of the Diamond Jubilee Celebration Lecture Series. We have started off this lecture series by a curtain raiser lecture from Dr. M.S. Veliyathan on 16th March 2023. Today, we have another eminent scientist amongst us. It's with immense pleasure I would like to briefly introduce our guest speaker, Dr. D. Sreenivas Reddy, who is the director of two very important Indian institutes, CSIR Associated Indian Institute of Chemical Technology, Hyderabad, as well as CSIR Associated Indian Institute of Integrated Medicine, Jammu, where he is also holding the J.C. Bose Fellowship of SERP, India. He received his PhD in 2000 under the guidance of Professor Govardhan Mehta from Hyderabad University, followed by postdoctoral training at the University of Chicago and later at University of Kansas. He then took up independent career in the pharma industry such as Dr. Reddy's Labs and Tata Advinus, followed by a senior scientist position at CSIR NCL Pune. Overall, he has 20 years of research experience after post-PhD in organic, medicinal chemistry, and drug discovery. He has authored almost 120 publications, and he is an inventor in almost 35 patents. To highlight some of his research activities, in his 7 years of pharma industrial experience, he has led drug discovery programs, and one of the molecule, licogliflozin, discovered by his team in industry, is currently in human clinical trials phase 2, where he was a project leader. He is involved in total synthesis of more than 35 natural products of biological importance. He has acquired skills in designing novel small molecules and lead optimization as a part of drug discovery. He has discovered an anti-diabetic molecule in collaboration with industrial partner Shantani, which is at advanced preclinical stage. He has two out-licensed technology developed by team at CSIR NCL. He identified silinezolid with high brain is to plasma ratio with a potential to treat brain infections. He identified novel mosquito repellents. He has developed a method to access enantiopure pheromones towards crop protection. He has identified novel clodosporin analogs as potent antimalarials. He has developed novel process routes for ivacaftor, a cystic fibrosis drug, lifitegrast, an ophthalmic drug, and azelaic acid, a topical cream for treating acne. He is recipient of various awards and recognition. Along with J.C. Bose Fellowship, he has received Shanti Swarup Bhatnagar Award and NASI Reliance Industry Platinum Jubilee Award for Application-Oriented Innovation. He is elected as a Fellow of Indian Academy of Sciences as well as National Academy of Sciences. He is recipient of Sun Pharma Research Award as well as Organization of Pharmaceutical Products of India Award. He is a nominated member of Scientific Body of Indian Pharmacopoeia. He is recipient of CRSI Bronze Medal for contribution to chemical sciences as well as CDIR Award for Excellence in Drug Discovery Research. He is one of the editor in the Bioorganic and Medicinal Chemistry Research Journal. His recent publications are seen in high impact factor journals such as European Journal of Medicinal Chemistry, Journal of Medicinal Chemistry, Journal of Organic Chemistry, Organic Letters, and Journal of National Products. With that, I welcome Dr. Reddy to MCOPS, Manipal. Now I request Dr. Reddy to deliver the DJC commemorative lecture entitled Role of Chemistry and Drug Discovery Process, a few case studies based on natural products. Over to you, sir.
Thank you very much, ma'am, for a kind introduction and elaborative introduction. So how many of you are aware of CSIR? Can you raise your hands? CSIR organization. Okay, quite a bit. That's good. That's a Council of Scientific and Industrial Research. So this is one of the largest organizations in our country. So it has about actually 37 laboratories across India, starting from Jammu Kashmir to Trivandrum. So we have multiple different labs, and we have different sciences like physical sciences, chemical sciences, biological sciences, engineering and information sciences. These are all different domains our organization works. And for all these organizations, this is one of the biggest organization. Always president is the Prime Minister of India. Whoever get elected, he will become the president of this whole CSIR. And similarly, next one is the Vice President is our Science and Technology Minister. He is always Vice President for this organization. And we have Dr. Kalai Selvi. She is a Director General of CSIR. That means she heads from headquarters. She is head of all the 37 laboratories. Like I am heading organization in IICT. Like IICT, there will be 37 laboratories across India. So she is the head of the whole organization, Director General. So just I want to highlight you that she is the first woman DG after almost 80 years of CSIR existence. So that's basically opportunity for all women students who can really head this kind of organization. She is our boss. Okay, and just to tell you that after spending, as per the introduction, after spending my PhD and postdoc, I came back and joined in Dr. Reddy's laboratories and also Tata Advinus. I worked in drug discovery area. Then after that, in 2010, November, I joined, there's a place called National Chemical Laboratory in Pune here. That's where I started my academic career in India. So that's where I began. Then I spent about 10 years there. I was lucky to get good students like many of you. After MSc, MPharm they joined, and they did, some of them they did PhD with me. And during that time we could do some good work. Then after that I got opportunity to become director of this institute. This is the Indian Institute of Integrative Medicine. That's located in the northernmost part of India, that is in Jammu and Kashmir. There are two branches. One is in Jammu, one is in Srinagar. So I was heading that one about almost like more than close to 2.5 years. Then recently, about 7-8 months back, Dr. Chandrasekhar, who was heading IICT, that is Indian Institute of Chemical Technology, that's here. So he became secretary of DST. DST is like CSIR. There is another department, Department of Science and Technology. That's again like under Ministry of Science and Technology, there are different departments like DBT you all know this one, Department of Biotechnology, Department of Science and Technology, CSIR. So he became secretary to that DST. Then this position fell vacant. Then that's how I thought I'll be one of the applicant. I applied and I got here now as a director of this institute. That's the Indian Institute of Chemical Technology. About 7-8 months back I moved here, Hyderabad. I also got opportunity to as an additional charge of this CDRI. I'm sure many of you are aware, right? CDRI. Can you raise your hands? How many people know before that CDRI? Yes, because you are all pharmacy students. If you don't know that, that is not good. So happy to see that you all know that one. That is the Central Drug Research Institute. I was there about 6-7 months. So basically, I was fortunate to get opportunity to interact with all, starting from National Chemical Laboratory, Indian Institute of Integrative Medicine, CDRI, and presently at Lucknow. So that's what my academic professional journey as far as CSIR is concerned. So what do we do at IICT? IICT it was established much before independence, that is 1944. It's almost 79 years of continued journey. So originally it was called CLSIR, then later renamed it as RRL Hyderabad, and in the year 1989 it became present name, that is Indian Institute of Chemical Technology. Right? What we do is we serve the society by creating outstanding knowledge base in chemistry and chemical technology. Although we focus on chemistry, chemical technology, but we have multiple departments, we connect with multiple different departments. You will see in my next slide. So here what we have in IICT is these are the departments. So like starting from organic synthesis and process chemistry, we work on natural products and medicinal chemistry and catalysis, fine chemicals, energy, environmental engineering, and oils, lipids, science, technology, and also analytical structural chemistry. This is one of the well-known department in our country. This is a beautiful facility what we have. And I was told that you also have MPhil analytics or some MPharm analytical they do, right? So this can be a good opportunity, sir, there actually. We have beautiful facility, that one. So we do have applied biology. I was talking about Professor Dhawan, Prakash Dewan. So he started that one. Originally used to be pharmacology. Now same department, we renamed it as applied biology. This one. This is one of the well-known department in the country. And we have process engineering, technology transfer, and fluoroagrochemicals, polymer and functional materials. Basically, although our major focus is on chemistry and chemical technology, but we are linked with all these multiple applications, multiple departments. That's what we do it. And we have contributed in many areas, starting from coal, fine chemicals, pharmaceuticals, polymers, lipids, agriculture, waste to wealth, even recent times, circular economy, chemical biology. So basically many contributions. I don't want to get into that one because I try to focus on different aspect today. So whatever we do, our connection is between healthcare, chemicals, energy, and environment. These are the areas all these departments work in a tandem. So just to tell you a couple of things, since it is also pharmacy college, that I'll tell you what we did in recent times. So we all went through these tough times during last 2-3 years, particularly during COVID time. So that time nothing was there. So not only like IICT, but many other CSIR labs, IITs, and other organizations in the country, they worked independently. Some of them they worked together, and to come up with whatever possible at that time. In the beginning, I don't know how many people you remember that one. Like we didn't have anything at that time, particularly in early 2020 when pandemic hit our country. So that time IICT worked extensively with some of these drugs because no drugs were there. So one of the drug along with Cipla they marketed, that is Ciplenza, that's basically favipiravir. The process development like with materials available in India. So developed and transferred to Cipla. That's where it is a marketed drug right now. And similarly other repurposing drugs also we worked. And in addition to that, what I would like to focus on this one is closely worked with the Bharat Biotech. I'm sure many of you had this short Covaxin, right? So Covaxin has many components. One of the component is that adjuvant, that's called TLR78 agonist molecule. That's one of the molecule, it is present in that Covaxin. It is in only micrograms, but again that material was not available in India at that time. So our IICT came up with some basically some synthetic route and came up with that, and they developed that process, and again they transferred to Bharat Biotech that material. So that's where we have a contribution in whatever we took today, Covaxin shots, some component of IICT is there. So that's where we feel proud about it and good about our contribution during these COVID times. And in addition to that, we also doing this clinical trials using these old drugs. One is a colchicine, one is a niclosamide. So when I was at IIM, along with the Laksai company and also the IICT, together we worked and we were basically started doing these clinical trials and with both molecules. That is for COVID treatment. So all the phase 2 clinical trial done. Now we are in the process of analyzing, and if everything goes right, we'll go for approval, or we may need to do one more trial. So at this stage, I would like to inform you all people this one. So that's a contribution from IICT during COVID times. So how do we get into IICT? How do we join? Because since many students are here, I'll tell you how you want to join. Suppose if you want to take up your career or if you want to enter into IICT, what are the options? You can join as a PhD in many groups, particularly relevant is chemistry and also biology and also some of the other related departments. So for that you need to have a master's degree. Master's degree means either M.Pharm or either MSc, M.Pharm or M.Phil you can choose. But only thing is we need to have fellowship. Without fellowship we don't have option at this moment. CSIR or UGC or ICMR, or you can also have Inspire fellowship to enter into PhD programs. You can also join as a scientist like me, PhD plus postdoctoral, PhD plus industry experience at different level. You can join in IICT as a scientist. We have our own, one of our senior colleague here is Dr. Chowdhury. So he worked several years in industry, decades, almost 3 decades, sir. Yeah, 3 decades he worked in different corporate sectors. Recently he was working in HPCLs. After 60 he joined as a scientist, is a distinguished scientist in our institute. That's kind of, you can join at any stage in the institute for different levels depending on your experience. So you can also join as a project assistant or associate if you have MSc, MPhil or M.Pharm or with the PhD some experience also you can join. That's what you can enter into. Even summer interns also we can join. Some of the projects also we can do together. So that's what I was telling you. That's, I think we already signed agreement, I think MOU. That's opportunity for all the M.Pharm and MSc students to do project together. Of course with the faculty, they can be like a mutual interest. If the faculty also has interest, they can also work together and they can also publish together papers. That's something, opportunity we can see here. Right? Okay, coming back to, so what I wanted to talk. So since pharmacy college, let me ask one question. What is drug? If somebody asks you a question, what is drug? So anybody can tell me simply? Yes, that is one thing. Yeah, any other? That's good answer. So there are many ways one can define, but I look at these are drugs. Okay, so I like this definition. There are many definitions available. Particularly, I like this definition of this US FDA definition. That is a substance intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease in human or other animals, and substances other than food. Of course, you always count it. Intended to affect the structure or any function of the body of human or other animals. This is what basically more scientifically we call it as a definition for this drug. And of course most of the drugs are small molecules. Many major more than 90-95% of the drugs today are small molecules. Again, there may be a little bit of teaching here, and I am sure you all heard about this one, is right? How do you name these drugs? Proprietary name, generic name. You all know, right? Yes, that's good. So like a Trimax, amoxicillin, this is antibiotic. These are the, again, I'm not getting into the details, but I'll just tell you few things. So what are the examples of drugs? I'm sure many of you know this one, all this acetaminophen, amphetamine, acetylsalicylic acid, captopril, diclofenac, and gabapentin, and this clomiprazine. All these drugs you all know, right? Okay. So these are some of the examples. Again, look at some of the examples of drugs. Natural products who went on became drugs. Reserpine, this is a neuromodulator, and galantamine. Again, these are all the alkaloids. So you know alkaloids, right? Yes, that's good. So this is one of the drug discovered in India from plants. That's isolated from this sarpagandha. That is a Rauwolfia serpentina. That's basically a small plant. You can look at antibiotics, streptomycin, erythromycin, cephalosporin, vancomycin. Again, these are all the, look at the complexity of the structures. You look at the complexity drugs here. They are all small molecules here. Look at this, how small this one, but at the same time you look at complexity of these molecules in antibiotics. Most of them are from natural sources, right? Look at this one, immunomodulator, cyclosporin, rapamycin, FK506. What is exactly immunomodulators? Do you all know this one? What do they do? Right. So those are for disease purposes. I can tell you one of the major contribution for this from these immunomodulators today is organ transplant. Without these drugs, there is no way we could have done any organ transplant today. So because when organ is transplanted from different person, our body doesn't cooperate. there should be some immunomodulator. That's where all this rapamycin, FK506 plays a role. Without those drugs, today I don't think we can transplant any organs. That's major contribution. Although they're all useful for, as such, if you have immunity like in a proper way, I think there are no drugs, no other diseases come into play, right? And look at the statins. Statins, again, one class of drugs, mavastatin, pravastatin, all these drugs. I'm sure you all know about these drugs. I'll not get into this, but I'll try to give you some kind of contradicting things. Like look at the structure, like how small the structure, isoniazid and rifampicin. Both drugs, this is a small molecule here, and you look at this complex molecule, rifampicin. So both drugs, they work for treating tuberculosis. Right? They may work in different mechanism, but at the same time, look at the how simple structure is this one and how complex this structure. Both are work for anti-tuberculosis drugs. Right? And look at this one. I'm giving you some example where you can appreciate how these structures and also their function. And look at these drugs. This is sofosbuvir. This is for treating for HCV. What is HCV? Hepatitis C virus. So HCV. So HCV drug, this is called sofosbuvir. So this is one of the, again, costly medicine, again $84,000, so expensive. And another drug called ivacaftor. This is for cystic fibrosis. This is another very expensive drug. This is about $300,000 US per patient per year, like basically per treatment. So that's how expensive you look at this one. Although if you look at the structure, it's not that doesn't look that expensive from chemical by looking at the structure, but whereas if you do real analysis, this is what $300,000 because one is how difficult to invent drugs. That is one challenging factor. Another thing is why this particular drug is very expensive, because very few patients available in whole world. If that company wants to get all the expenditure done on that particular drug, so you need to recover from only those patients. So because they are also business, right? They also need to get their money back. So that's where this particular drug became very, very expensive. This is one of the reasons. Okay, how drugs are discovered? Again, I know you all went through this kind of slides in many occasions, probably in your during the course of your study. Starting from target identification, went through all this process you get into, like phase 2, phase 3 NDA and also phase 4. And about cost is about more than $2 billion US, about 10 to 15% years it will take. But I would like to give some numbers here. Chemist like us makes about 10,000 compounds. Out of that, only 10% compounds will hit into this pharmacology safety compound, about 1,000 compounds. Out of only 10 compounds will go to clinical development. Clinical development means where you will get into human clinical trials. Out of that 10, only one gets a chance to become the market. Look at the statistics. You start from 10,000, you get into one molecule into the market. That's kind of statistics available based on different clinical trials outcome available. This based on this all the put together analysis. So, but I'll give you go through this kind of pictorial way so that you will understand better, you will appreciate better. And any professionally like driven drug discovery companies, what happens is once the target is identification, when the team is formed. Again, team comes from chemistry, like medicinal chemistry, biology. Biology also different again, pharmacology and pharmacokinetics and regulatory and different domains, and also process development. All the team is formed along with the project manager. Then they define that project goals and objectives, everything. So once the team is formed, when the goal is set, then chemist goes and makes a standard compound, and at the same time biologist goes and makes set up assays using standard molecules. When the assays are set, then chemist goes back and make molecules. I call it as a proprietary molecule here, because for academic setting it's okay, but in industry, if you want to make profit out of it, or any company if you want to run drug discovery company, you need to have a proprietary molecule. That means patentable molecules you need to have. So chemist goes and make a patentable molecule, then in tandem with in coordination with the biologist, they screen compounds, and using again various screens available medium throughput, high throughput screening. Once you are through with this one, then you go to in vivo efficacy, again based on the disease. Suppose if you are looking for diabetes models, you can DB model, OB model and other things you will see. And once you are through with this one, then you go to extensive safety studies. That's where most of these pharmaceutics and other things play into the picture. Once everything is through with this one, then you go for IND application in regulatory authorities. IND application in regulatory authorities means it is like US FDA. Like in India, what we have is a DCGI, the Control General of India. So we go there, we submit application, and they review in a month time. Once they are, when there is no objection from them in a month time, then you can go and dose in humans. So that's where you get into phase 1 clinical trials. So what is phase 1 clinical trials? Where you take healthy volunteers, right? So you check the safety of that molecule, whatever you made this molecule, went through this cycle, safety of that molecule. First you do single ascending dose, then you do multiple ascending dose. It's easy to remember for you, SAD, SAD, and similarly MAD, MAD. So based on that you will know what kind of dose is safe in healthy human volunteers. So once you are through with that phase 1 clinical trials, then you go to phase 2. In phase 2, real patients are taken there. So that means suppose I am giving example of diabetes patients are recruited here, and you do that based on the safety dose, accordingly you calculate and you do this dosing here for human patients. So once you are through with this one based on the effective dose, after finding effective dose, then you go to phase 3 clinical trials. So phase 2 and phase 3, the difference between those two are like only scale up, and also you do multi-centric places, you do different centers, you do that one, different countries also you do this one. So once you are through with this one, then you go to registration. So that's where again you get into regulatory authorities. So regulatory authorities, they make domain expert group they have, then they review that application. Once they are comfortable or they are satisfied with all the data, then they approve that drug. So once you approve this drug, then let's get into clinics. So that's kind of what I am trying to say. It's a long road starting from here. There are so many hurdles, but ultimately to reach there, at least it will take 10 to 15 years average time to discover a new drug. Right? For any purposes, I look at is like not only that, many of us look at early stage is very, very important. Choosing the right molecule is important. That means a discovery stage, if you find a right molecule, then easy to go forward up to clinics. That's where you need to focus more on that one. So here is a funnel I look at. So NC is that new chemical entities are made. It will put through this funnel. Primary assays, secondary base assays, selective assays, in vitro ADME, acute in vivo models. Again, depending on disease, you choose those animal models. Then again, advanced disease model. Then of course you need to do toxicity parameters you need to check. Once you are through with this one, you get into the developmental candidate. So during this process, so wherever you fail in this process, you need to go back to make new molecule or design a new molecule and come up with a new synthetic variation or new synthetic using new chemistry. You make new molecule, changes accordingly depending on what is the problem here. So that's where what I am trying to say is chemistry, and particularly medicinal chemistry, plays a major role in drug discovery process. So that's what we try to connect always. Chemistry is very, very key component or very important component of drug discovery process. Okay, how do we start as a chemistry starting point if you want to discover a molecule? So library screens, you can do large number of drug-like molecules. It's available for testing. There are many commercial sources also available now. And you can also do literature leads. So there are literature available, and most of the academic institutes, they do literature analoging. Something known in the molecule, known in the literature, you take that molecule and do analoging. That is one way to do it. Only thing is a patentable space is very, very challenging from the known literature molecules. And also analoging of ligands, substrates. Again, another important starting point is structure-based drug discovery, structure-based drug design. So here, if the target protein structure is available or if it is known, that is the best way to start any drug discovery program. So where you will have a real logic to like basically how to modify, how it is binding, which amino acids are important, which structure is important. That's where information is very, very important. Of course, in the absence of 3D crystal structure, protein crystal structure, you can also do some of these homology models that can be developed using known ligand. This is another way to do it actually. I'm sure some of you must have learned all these things, right? Did you learn or no? Yes, good. So another one more way is like medium throughput screening, we call it. So you can also use one target class based libraries, and also in particularly natural products like compound, that is very, very important, and screening of fragments. That's one of the things we try to do. So in my own research group, we focus more on natural product drug discovery. So that's what I'm trying to bring the connection from drug discovery background, and that's where I want to talk about a little bit about what we do in my research group. After spending few years in industry, when I started my career at NCL Pune, so we started working on two areas. One is total synthesis of natural product. Another one is application oriented, some of these societal application, particularly crop protection, insect repellents, and also medicinal chemistry towards drug development. Of course, we always work on some of these areas, but it's not very focused on that. So antibacterial, anti-parasitic, anti-diabetic, and also anti-inflammatory. Some of these we have been working on last decade also, almost like now 12, 13 years. So why natural product? Why did I choose that one, natural products? So in today's market, whatever available drugs, out of that, about 40% of the drugs, 40 to 50, close to 50 actually, 50, close to about 40% of the marketed drugs are related to natural products, either directly or indirectly. And out of that, 80% of the commercial drugs which are derived from natural products require synthetic efforts, because most of the drugs, they are not available, most of the natural products, they are not available in good quantities. Another one is they don't become drugs as such. You need to make some changes. Some of them are toxic, some of them are not soluble, some of them have different selectivity issues. Because of that, you need to modification, and you need to get supply, more supply. That means you need to have synthetic feasibility to make those molecules. So for that purpose, and also in my why another strongly I believe is students who work on this total synthesis, they get exposure to variety of reactions, because they need to do multiple reactions, different steps, different exposure they get. And also they are also challenging, particularly they become very strong mentally, because most of the time unless you complete the natural product, you don't assume that that is a completed target. So that's where total synthesis offers structural modification and supply of natural products and structural simplification. I'll tell you a couple of case studies what we do in my research group. Again, just before that, again these are the natural products went on to become drugs. And here one example I am giving you, this Reserpine. This is a plant, I was talking about it. This is when I was in IIM Jammu. So in our farms we used to have this Sarpagandha. This is the plant, Rauwolfia serpentina. So this is, it's a good connection with India. So it was isolated in 1952 from these roots, dried roots of this Rauwolfia serpentina, and also many well-known scientists at that time, Dr. R.N. Chopra, Dr. Mukherjee, Dr. P.C. Ray, all this, Dr. Gannath Sen, Karthik Chandra Bose, Siddiqui, all these very big shots at that time. They worked on this plant. So another connection I have with the natural product is when I was working in Advinas Pune in collaboration with Novartis, we were working on this program. So here is a connection, natural product connection. Natural product, it was, this is called florizin. It was originally isolated from this apple tree, and it showed anti-diabetic effects. Anti-diabetic means it's basically it reduces sugar level in plasma. So through structural modification, so they made this compound called T1095. So this T1095 and if you look at original compound, both are having this O-glycoside. So you all know, right, what is O-glycoside? Basically glycoside linked with this moiety, aromatic moiety through this oxygen. O-glycoside we call it. So both are O-glycoside. In our body we have glycosidase enzymes, so that has the capability to cleave those, so that will become unstable. So then after few modification it became this dapagliflozin. This is a C glycoside, so which is stable to O-glycosidase enzymes. That's how it became now. This became a drug, dapagliflozin, and similarly we have few drugs like canagliflozin. They are all marketed drugs. During that time when I was working in Advinas Pune along with Novartis, we invented a molecule that LIK066. That is now it is called as the licogliflozin. So that time I was a project leader in industry. It is presently in phase 2 clinical trials, and also it is being tried for NASH and also PCO patients. This is basically, I call it as a kind of good opportunity for us, and scientists or chemists like us who design molecule, and also with the help of big team, the chemist we made, and finally it is in human clinical trials. That's a big satisfaction for medicinal chemists, right? So, okay, these are the natural product we synthesized in my research group after joining at NCL Pune. Again, these are all these compounds are synthesized and published, and many of them are first time synthesized. That means no synthesis was there before that. Using all these compounds originally isolated from natural sources, and also some of them are marine sources. And what we did in our laboratory is synthesize them in some good quantities, and also made them some of the analogs. And finally we are making it towards medicinal chemistry program towards lead optimization. And in that case that is what I wanted to show you, couple of case studies what we do in 2 cases, because it's difficult to talk about many of these drugs, but I will show you 2 cases. Here is a cladosporin. It was isolated in 1971, long time ago. This is a fungal metabolite. And it why we became interested recently, because recently this target was found. Although it is anti-malarial natural product, it showed good activity against Plasmodium falciparum, but recently they found that it is working through lysyl tRNA synthetase. That is one of the target for for this malaria, and also good selectivity when compared to human counterpart. It's about 100-fold selectivity. And although a couple of syntheses are known from Shi and also Mahapatra, there are 2 syntheses are known, but what our interest was slightly different when we took up this project. Because of this potent malarial activity and also selectivity and novel mode of action, particularly this lysyl tRNA synthetase inhibition, and lack of systematic SIR. So that's where we thought we'll take up this project. So look at this molecule. Carefully look at how many chiral centers are there. How many chiral centers? I'm sure chemistry people are there, chemistry students are there, organic chemistry students. How many chiral centers? 3, that's good. 1, 2, 3. 3 chiral centers. So how many isomers are possible? That's good, very good. 2 to the power of 3. Right, 2 to the power of 3 means 8 isomers are possible. So we thought that let's make 8 possible isomers. Then look at which one is a better molecule out of 8 molecules. But making 8 molecules is not that easy, because all are very close structures. Only difference is up and down, up and down. So our plan was to make all the 8 possible compounds. So that time one of the student, like you, only one student, he did MSc chemistry from IIT Chennai, IIT Madras, Pranay. So he joined with me that time. So I put this problem in front of him. Initially he was reluctant and also shocked how to make all these compounds and how to characterize. Of course, that's where we give them training. So plan was to make all the 8 possible, because of 3 chiral center, 8 isomers are possible. 8 we need to synthesize all the compounds and subject them to biological assays like enzymatic, parasitic and selectivity assays, and finally come up with the best molecule out of this. What I am trying to do from this exercise is which is the best fit for this lysyl tRNA synthetase, which is better binding, ultimately that we can become as a part of lead optimization, which stereo structure is important. So we all know about structure activity relationship, SAR, right? Here what I am trying to do is additional component we are adding SSAR, that is stereo structure activity relationships. That's what I am trying to do here. When the student took up this project, so we started with the synthesis. Again, I am not going to go through all the details, but I will just give you the glimpse of what we did is. So this is a dimethoxybromobenzene, and we went through a couple of steps to make this allyl derivative. Then you do a couple of more steps to get into this fragment A. This is one portion of that molecule what we wanted. And similarly for the chiral moiety, we started with the S-propylene oxide, again opening of this epoxide and protection, and again do ozonolysis and Wittig, and again reduction. You get these two isomers here, and again you get into deprotection of this compound, you get this mixture of these compounds. Then finally you treat with this particular reagent, palladium reagent, you get to this fragment B. So we have fragment A and fragment B, both, right? Once you have this one, then you combine both. There is you all heard about this metathesis, right? There is a ring closing metathesis. Have you heard about it? So there is, in organic chemistry, there is if you want to combine, like suppose if you have two end olefins, if you want to combine and make that one olefin, that's called ring closing metathesis, making rings. This we call it as a cross metathesis. Basically between two molecules, two olefins, you are combining and making it as a cross metathesis. So we did this one, then we get these two compounds, we separate these by column chromatography, and again osmium tetroxide mediated dihydroxylation we do, like basically hydroxylating double bond. So we do this one of the hydroxy, what happens, it cyclizes with ester, another hydroxy will be free. Then you do some this thiocarbonate route, and finally you remove that one, you get to this cladosporine. Like similarly, different isomers gave us different things. Again, basically what we made is clado1, clado2, clado3, clado4. Made fine, theoretically all looking fine, but when you look at NMR, they are all very, very close by NMR. It's very challenging for us to distinguish between them, although we know what we made, but if you want to convince reviewers, or if you want to present to public, then how do you make sure that? You need to have confirmatory structures. So we made 4 here. Similarly, instead of S-propylene oxide, what we did is we used R-propylene oxide. With R-propylene oxide, we made all this clado 5, 6, 7, 8, like all remaining 4. So we did extensive NMR, and we are convinced, but at the same time we wanted to make further confirmation. For that, what we did is we are also a little fortunate to get crystal structures of all the 4 compounds. So we have crystal structure with absolute stereochemistry, we have now 4 compounds. Since we know this one, we also know the optical rotation of other molecules. We compared all the NMRs and everything, we found that all the 8 isomers are perfectly matching, whatever required. So once we have this 8 compounds are made, then entire library subjected them to biological assays. One is a thermal shift assay, that where you will find that how closely or how much affinity with the ligand towards that target protein. That's what we try to measure using this TSA. Then after that, you need to check how much inhibition in parasitic growth, where you can also calculate EC50 particularly against this Plasmodium falciparum growth inhibition. Then after that, enzyme inhibition assay. After all these things, what we wanted is like what is the active isomer. Here is the result what we have. So out of 8 isomers, 2 compounds, clado1 and clado5, became like more potent. Like it showed actually like and also it closely bound with the protein, target protein, lysyl tRNA synthetase. We got co-crystals and we co-crystallized these both the compound clado1 and clado5. The difference is only here, this particular stereochemistry. So everything else was not active. Other 3 compounds are moderately active, and remaining 2, 3, 4, 6, 8 compounds are like non-potent, like they are not active compound. So what we try to do with this exercise is all these 8 isomers, subjected them to biological assays. We found that 2 compounds are the best compounds. So we try to show that in drug discovery, stereochemistry is very, very important. Although it is not that first time we are proving, but using this particular scaffold, this particular enzyme target, we showed once again that the stereochemistry plays an important role in drug discovery process. So once you are there, then next what? Then we need to of course, we also made scale up, but let's not get into that one. But what else we did it is, we took this particular scaffold and we went through this lead optimization process. Several analogs were made, and we finally came up with the new lead molecule at this moment. What we have is, after systematic CSIR, this particular hydroxy additional here we added hydroxy addition that has additional interactions with some of the amino acid around that moiety. So that's where we have now a good molecule at this stage. That's one story I wanted to tell you. And the second story is, this is related to hunanamycin. This is again one of the molecule isolated from marine sources. Look at the structure, this and this one. The structure, some similarity is there, particularly this sugar moiety. This ribose moiety is very similar, including basically all the stereochemistry of ribose here and here, both places are same. Right? It showed anti-salmonella activity, that is a 4.8 microgram per ml. And why are we interested in this target molecule? There are a couple of things. Because this is although in India we don't call it as a very serious disease, but it causes, in overall global burden is about 600 million foodborne illness occur, about 4.2 lakh people death occurs annually, and major risk in particularly low income countries. In our country or in underdeveloped or developing countries, we don't have full data, but at the same time it is very, very serious problem. This food poisoning illness, foodborne illness. So another interesting factor why we wanted to work on this target is, this works through riboflavin synthase inhibition. So riboflavin synthase is one interesting target. So riboflavin synthase is only found in bacteria, not in humans or higher species. We get riboflavin from food, whereas riboflavin in bacteria they have to synthesize. They have this enzyme, riboflavin synthase enzyme. So if you selectively block that enzyme, you can selectively kill the bacteria without harming humans or higher order species. That's the idea. That's the idea behind going after this target. Again, hunanamycin. If you look at Lipinski rule of 5, have you heard about Lipinski rule of 5? Yes or no? Yes, good. So Lipinski rule of 5, when we calculated for this particular scaffold, so it found that there is perfectly matching. There is no violation with respect to Lipinski rule 5. So when we are choosing this molecule, we also look at some of these parameters. Not that we are very strict on Lipinski rule of 5, where it can become like more possibility to become a drug if you follow Lipinski rule of 5. So there is no violation. That's where we became more interested on this particular scaffold. There was no synthesis known at that time. Very small quantity was isolated. Less than a milligram it was isolated originally from marine bacteria. So we wanted to make that compound and go off, like synthesize and lead optimize the scaffold. For that purpose, what we did is, we took this particular starting material. This is easy to make. This corresponding diamine, if you take oxalic acid or ethyl diaxylate, if you heat it together, you will get solid crashes out. You get good solid. Then you do prenylation using this particular reagent, cesium carbonate. Although yield is very less because of, there are two possibilities, right? Here and here, both places can alkylate. So using some optimization, like lower temperature, and also using this particular base, we get this 42% yield of this particular product. So once you have this one, then you subject them to Friedel-Crafts alkylation. So using aluminum chloride. So we get this tricycle compound. Once you are there, then you do with this allyl derivative, and finally osmium tetroxide mediated dihydroxylation, and remove this protecting group, you get to this hunanamycin. So that's about 80 milligrams we synthesized this compound at that time, almost like 4-5 years ago. So we made this compound, and interesting thing is, originally it was isolated less than a milligram, but we could make more than 80 milligram. That is a good sufficient amount for taking forward and doing further studies. So again, we were not very happy at that time. So look at this one. This is a starting material cost is about 25 grams of this material. It costs about $450 US. And we also need to do several steps to make this compound. And similarly, if you look at other side, very similar structure, there is riboflavin. That cost of 25 grams is $32 US, very cheap when compared to this one. So then we thought that maybe nature is making starting from this riboflavin. Why can't you synthesize starting from here? So then Rahul Shingar, who was working at the time in this project, so we both discussed and came up with the new synthetic route making this compound. How did we do? We started from riboflavin and subjected them to this sodium hydroxide and H2O2 treatment. You get this removal of this ring, you get to this product. Once you are there, then prenylation using potassium carbonate, you get to this N-alkylation. And once your N-alkylated compound is there, again subject it to aluminum chloride mediated Friedel-Crafts reaction, you get to final compound. So look at this one, 0.8 milligram original isolation. Now we can make 10 grams of material, this one, without much difficulties. 3-step sequence, gram scale synthesis, no column purification is required, only washings are required, and overall is about 48%. It's a good purity also, 98% purity after washings. So that's where you get good satisfaction when synthetic chemists when completed this target in a good scale. So I will also tell you, so summer train is comes to my group, particularly when I was in NCL. So I used to give this project to them, give the starting material, this riboflavin, that's very cheap, you can buy, and give some of these reagents, they make this compound. So like without any proper training, you can make grams of this material. So students used to make this one. So if you are interested, you can also start as an experiment, like a practical experiment, this one. This is so easy to make these compounds, this one. If you have riboflavin, if you have these reagents, you can make this compound. So that's one of the good contribution from what we made from our laboratory. So after that, what is the next step? Although we assumed that this is working through riboflavin synthase, we need to have some proof always. So we collaborated with Professor Macmillan from USA. So what we did is a penultimate step. We incubated in E. coli along with riboflavin synthase and different concentrations. So what we showed result is here is a decreasing concentration of hunanimycin. So it increases the production. So basically what we try to show is that this is going through riboflavin synthase inhibition, because when you are decreasing concentration, here it is the product is increasing. That's basically inversely proportional. That's where we know that it is working through riboflavin synthase inhibition. Once we know this target, like indirectly we proved this target, then next we also went after this lead optimization, structure activity optimization. What we did is we took all these different substitutions here and different polar groups. We varied. I'm not going to go through all the details. We made more than 70 compounds which are looking like a hunanimycin, then optimization, then we get to the final compound. This is a 5,4,8, this is the latest compound what we have. So these are some of the attributes we can make as a part of a structure activity relationship. It showed excellent pharmacokinetic properties, and also it showed very safe. Basically what we showed is toxicity. LD50 also we determined it's more than 2 grams per kg. So that's again very good molecule to take it forward. So we are also doing this in vivo experiment with this. We are also looking at it can be used as a food preservative, because salmonella is one of the serious problem, particularly in the meat industry. So we are trying to explore that option also. It's a huge market potential. This is what we recently published some of this work in the literature. So last part, next couple of minutes. So I thought I'll show one thing which is not relevant to pharmacy or drug discovery, something different, societal application. So we know this grapes, we all eat grapes, right, draksha. So in that you see that white powder, most of the time we see that white powder. That white powder, if you take magnifying glass and expand it, this is what these are creatures. They are all mealybugs. Sometimes we eat also those, right? So those are those creatures. Mealybugs they call it. So this is not only problem for grapes, it is also problem for cotton, mangoes and pomegranate, and papaya also serious problem. Papaya also you see that white powder. So right now there are different ways to take care. One is you can also release this, this is called beetle, this is ladybirds. So if you release them in the crop, they go and eat that white powder. That is one way to take care. These are all safe creatures, they fly out when you take there. So then other one is you can also wash with pressure pipe. If you wash with pressure pipe, all this you can remove that, but at the same time, after some time again it will grow from it will crawl back again. That is again another problem. Right now most effective and also widely used method is insecticides. insecticidal soap, you can use it and you can take out, you can take care of these mealybugs. But that has always problem, right? Toxicity issues, long-term effects, and if you want to do export, you need to take care of this residues, all those problems are there. So in that connection, there is a new technology coming up, that is the use of sex pheromones. So what it is, so in this case, what happens is mostly in the case of male, in this case of male bugs, female mealybugs, they secrete some pheromones. They are very attractive to male mealybugs. So you take small amount of pheromone quantity and put it in the trap. This is called as a delta trap. This is a sticky sheet. You keep small amount of that pheromone there and leave it in that fields. So what happens is all male mealybugs, they think that female is sitting there. So they go and they get attracted and they get trapped here because that sticky sheet is there. They get trapped. You take out that sheet and you burn it and also destroy that one. So in this process, you are destroying all male mealybugs. Male mealybugs. If you do 2-3 cycles, you are completely removing that all male counterparts. So there is no reproduction there. So that's how you can take care of those mealybug problems. This is one of the recent technology coming up, and many people have started using this one. This is very, very highly sensitive, highly selective, and also very safe method because no toxic materials we are using here. We are only using that natural pheromone isolated from female mealybugs. So in this connection, one of my students joined at that time, Remya. So we gave I gave this problem, I discussed with her, we gave this problem to her to make these pheromones and test it in fields. So for that purpose, we synthesized both these compounds, plus pheromone and minus pheromone. This is originally isolated from female pheromone, but that will be available in microgram. That won't be sufficient to treat, sufficient to use it for large field trials. You need to make synthetic material. That's where chemists like us, we make these molecules and basically she synthesized these two molecules, plus isomer and minus isomer. And after doing this one, then we collaborated with University of California Professor Miller. So where we sent 1 milligram of each compound, 1 milligram of plus isomer, 1 milligram of minus isomer to test it in their field trials, how good, how attractive they are all. So what they did in USA is this is a 5 microgram, this is 5 microgram plus isomer, 5 microgram minus isomer, and this is a racemic material. That means 5 plus 5, 5 plus 5 minus. So basically 10 micrograms of racemic material. They tested and they found that one of the isomer found to be superior when compared to minus isomer. That's got really excited us, and we basically that professor and we got excited about it. We published a paper and patented. After seeing these results, another group from New Zealand contacted me to send some material because this is very, very interesting pheromone. So then we sent 1 milligram each, 1 milligram each, 1 milligram of plus isomer, 1 milligram minus isomer to New Zealand. So where I closely work with Andy Trudel. So what they did is they tried the similar trials what we did in California vineyards, these Hagbees, that's very famous vineyards there in New Zealand. They found similar activity, in fact slightly better. They used a 10 microgram and 20 microgram of racemic material. So they found that they showed better results. Look at the two different geographical locations. One is in USA, California, another one is in New Zealand. So both places it showed excellent results. With small amount, you can do like a half hectare with 1 milligram of sample. So that's where we are very, very excited. Then what we thought that we'll try it in India. So Pune, particularly nearby Pune, there is a Nasik area. That's where we tried, but we could not find encouraging results. Although there were some results, but there were some mealybugs we could attract, but it was not like this one. There could be two reasons. One is probably we don't know that how to do that experiment. Basically, we don't have that entomology expertise. And another could be that species may not be there. That same species what we found in New Zealand, what we found in America, California, may not be the same species what we have in India. So that's what we are trying to find out what exactly the reason. But in the meantime, we also have some, like basically another request came from New Zealand team only. They want to do it in Australia. That's where, so we need to make some of this compound and send it to them. That's where we are at this stage. So what I tried to tell in last 40 minutes also, So we work on application-oriented organic synthesis in my research group, and in particular on natural product synthesis towards application of human welfare, particularly towards identifying new scaffolds and new leads for different diseases. And we identified a few leads, and hopefully they can become useful in the near future. And in addition to that, I have my own satisfaction that students who trained in my research group, they get a feeling, or at least they are trained in the direction that they are working for the welfare of human. Although it may not be medicinal chemistry, it may be like other applications for societal problems. So here is my research group. So they are all very hard-working students. Again, they are all joined like you, only like after MSc, MPharm, they came to my group. They started their career. This is my first set of students when I joined in NCL Pune. So they are all real pillars for my research group. They started, and all these students completed their PhDs, and many of them are started their careers, and few of them they are looking for opportunities now here. But this is not my current research group at IICT. So they are also last couple of years, because of my movement from Pune to Jammu to Lucknow to Hyderabad, they are also scattered. But right now we came to closer, all are now together. We are together in IICT, but hopefully we will continue to do some good work. So that's what I wanted to tell you all. Again, these are all my funding agencies. Any research you want to do, you need to have a good funding. So these are the government agencies what we get funding, like DBT, DST, Minister of Earth Sciences, CSIR is my parent organization. In addition to that, what we get good success is if you have a good collaborators. Although I am a chemist, but I always have connections with, like basically collaborations with the different biology groups. Without their support, I don't think we can create value for those molecules what we made in our laboratory. Again, I would like to thank my original organization NCL, IICT and IIIM, where some of the work is being carried out. And thank you all for your patient listening.
Thank you, sir. I think that was very informative lecture, and I hope that all our students and faculty are benefited from it. On the occasion of Diamond Jubilee celebration and as a token of gratitude and appreciation, I request our principal and vice principal to escort Dr. Reddy onto the dais for felicitation. It's time for vote of thanks. My heartfelt gratitude to the chief guest, Dr. D. Sreenivas Reddy, who took time off from his busy schedule to be here with us for presenting the commemoration lecture. I thank the management for providing the facility to organize this event. My thanks to our principal and vice principal for gracing the occasion. I thank our dear PG students who have come in good number, and the head of the departments, the research scholars, faculty, for sparing their time during this lunch hour and for being here to listen to the guest speaker. Last but not the least, I thank all those who are involved directly or indirectly for making this program a successful one. Thank you one and all once again.